not a journal conference. It is being recorded and the live webcast is posted to the Ministry of Education. We are very adherent to the time. We should start at 10 o'clock. 5-10 minutes is fine, but we start at 10 and 2 lectures before 1. 10 to 12 there will be lecture and again there will be a content. Those who are taking attendance, Pushpa, take attendance at 10 sharp. Those who are not coming will not be allowed for entire day to attend. If you are attending, you have to come at 10. Otherwise, you have to leave the whole session and certificate will not be provided.
You have to come daily. If those who are not coming daily, the certificate in the last is not allowed. Your attendance is mandatory in the lecture and the practical session both. So be very strict on the attendance at 10 o'clock and allow only these people to attend throughout the day. If someone is coming, ask me to meet me and then I'll see what reason they are doing. So, okay, I hope you will attend it seriously. I again invite Professor Chodoro this session, lecture 1 and lecture 2. After this lecture, we'll have a short break and then second lecture. Professor Chodoro is yours.
Okay, good morning everyone. So you managed to rest a little bit? Yeah, I hope. Well, we're going to continue with the story and trying to go to a little bit more details on what we was doing actually yesterday. So if you think about yesterday the work, what we was doing in the practical, he is explaining the procedure again and then maybe some doubts that you have will be fixed better. Okay, first of all, we have the commercial samples. We have this pickle, then we have the ducky, and can you see the plate in the bottom? Okay, so you have this bottom line here. You have the bottom line. Then we spread, we homogenize the sample, because we get a liquid sample, so we don't need to homogenize. We scratch from the mango to make it easily. And since we're not caring about the number of the cells we have in the samples, we was directly spreading with the loop. So we're putting the samples in the top, then we cover with a second layer of the agar, and the plates are incubating now. I hope will be some individual colonies today, and we're going to apply a third level that will be here. We're going to cover with another layer of dagger, so we wanted to see something like that today. This is what we want to see, because this means this colony here, this one most probably is producing something, and not this one, because if it was this one, the zone will be like that. So something what we want to see. I hope we will see, because no one can guarantee. But this is the point. So again, here it's explanation on the entire process again. You have the samples, sorry it's in Portuguese, but I'm explaining now. Use samples, you can you can mix with the physiological water always. Then you can make a dilution, then you can plate on the agar, then you cover with another layers, then you incubate, then you observe from the incubation zones if you have. Then you pick one colony from the incubation zone, you go to the culturing them in the liquid cultures, and then you're going to check catalase, gram coloration, and then some other tests, but it's coming from the way as well. There's another way. This is the way when you try to explore the new producer, if you have the raw product. In case you have the real culture, already you have your culture collection in the laboratory, and you want to explore if some of the culture collection is already going. So you're doing it much more simple. You make agar with the test organisms, and you spot just a small amount, could be 10 microliters, could be just one touch with the loop of the culture, pure culture that you want to test. You incubate and you observe. If you have something like that, the culture growing and have the incubation zone, there is a chance to be. But this is only in case if you have already pure culture. So you're going to observe something like that, I hope, tomorrow, because after you see the test organism is growing, and here most probably it's a bacteriocin. Very important point on this experiment is to go to have the pure culture, you know, the standard microbiological procedure. You need to be sure that you have the pure culture by streaking on the plate, picking a colony, streaking again. It's taking time, but there's no another option, because if you have contamination, you can never work properly. And then freeze dry, if it's possible, or keep in -8. It's always important to keep more than one Eppendorf, because you're going to keep it in -8 in the presence of glycerol as a career protector. But it's important to keep at least 5 or 6 tubes, because sometimes happens something. Even it's good to put it in the 2 different freezers, in 2 different places. I was working in South Africa, and one laboratory took fire. Everything was on fire, and they lost entire collection. This is the good point. Very often what we do, we keep it in our another building laboratory freezer. We're just taking one freezer to a different building, and we keep all the collection in duplicate. Because if you lose everything, imagine you're doing a PhD on the last year of the PhD, and you lose the collection, you lose the bacteria. All your PhD is gone. You cannot defend, because you cannot continue working. You need to start from scratch. But then it's important to check if this bacteria is really producing a bacteriocin or not, because as you remember from yesterday, this could be an acid, this could be diacetyl, this can be a Reuterin, this could be even antibiotic if it's some strain of bacillus they're producing antibiotics. So we need to do the check for the bacteriocins. Normally the check is quite simple. We need to have the supernatant, so you centrifuge, you obtain the supernatant. Then you need to correct the pH. Why? Because correcting the pH, you're excluding production of the acids. In this way, you're sure that what you will detect as activity is not going to be acid. And we need to do the treatment for 10 minutes at 80 degrees Celsius, because in this way, you can destroy, if it's any hydrogen peroxide produced, you will destroy it. And as well, if you have any sensitive enzymes, proteinase, will destroy them. Because some of the lactic acid bacteria, they're not very typical. They're not good producer of proteinases, but some of them produce. Imagine you have the bacteriocin, it's a protein, and you have close to him a proteinase enzyme, what will happen? Enzyme will destroy the bacteriocin. And then we spot 10 microliters of the surface of the plate and incubate and observe for such activity. Now it's an entire method explained more in detail. So you culture your bacteria. You can incubate. Well, in this case we put 30 degrees, but if you work on the strain that you want used as a probiotic, you need to pay attention. What kind of probiotic are you going to use? If you're going to use it for human, are you going to use for the chickens? Are you going to use for the fish? Okay, someone veterinarian here? No one with veterinarian background? What's the temperature of the chicken? Yes. Please, just try it. Are they warmer than humans or they're colder? Warmer. They're about 39, 40. So if you develop the probiotic for the chickens, you need to be culturing every time in 40 degrees, because this bacteria, you want to know how it will behave when it's in the body of the chicken. And if you're doing a probiotic for the fish, it's any fish living at 40 degrees Celsius? No. It's like 25 maybe, but even lower. Depends where the fish will be growing. If it's in the Amazon, in the tropical area, obviously maybe 30, maybe not really, maybe 25, 30. But if you have the salmon in the Norwegian area, it will be like maybe 15. So you need to check the temperature that is objective of your work. You grow, you centrifuge, you obtain the free supernatant, you can correct the pH, you do the treatment to destroy the proteases and as well destroy the hydrogen peroxide. And then you need to spot on the plate. This plate, when you use for the bacteriocin test, we prefer preparing them with a soft agar. So in this case, since I work with Listeria monocytogenes, I'm using the BHI with the agar, and then I inseminate Listeria inside. Normally, we need to go to the level of 10 to 5, 10 to 6. So there is a practical tip how we can do this. It's more or less 1 microliter of Listeria culture for 1 milliliter of the agar. And in this way you're reducing already 3 logs down. So later today when we go to practical, we need to pour this third layer on the plate. So we're going to do these dilutions. And it's a very practical way because, okay, if you prepare 1 plate, you can make a 100-time dilution. You put 1 milliliter of this diluted Listeria, then you pour in the top agar, you mix the plate, it's fine. But if you need to make 50 plates or 60 plates, it's not easy. So rather make this proportion, 1 microliter of the overnight culture of the test microorganisms with for each 1 milliliter of the agar, you mix in the bottle, you mix the bottle, you pour the plates. And then you sure that you have the same amount of the Listeria for all the plates. Then you spot 10 microliters of the supernatant in the top, already treated. Then incubate and observe for the presence of the zones. And if you have the zones, bingo, it's working. If it's no zones, let's start from the beginning. We need to look for another strain that can be produced. Okay, now the question is, how strong is our bacteriocin? The way of measuring the activity of the bacteriocin is the same way that people measure activity of the antibiotics and the enzymes in the units. So for the determination of the units, we're using critical dilution method. So now it's going a little bit more complicated because I need to explain this method. So you're taking normally the ELISA plate to do the dilutions because it's easy. You work with a small volume and you can do it in the Eppendorf's as well. So you distribute to each of these places a phosphate buffer because we need to be sure that the protein will feel the stable at the presence of the environment. Normally the proteins are very well having good stability in the phosphate buffer with pH 6.5. Could be 25 up to 100 millimolar, no problem. It will be okay. So you're putting like the same volume here. Let's say you can put 100 microliters for all of them. Then you take your bacteriocin, your supernatant, and you're putting the same volume. If you put it 100 microliters, you take 100 microliters cell-free supernatant, mix, pass 100 here, mix, pass to the next, mix, pass to the next. So you're doing 2-time dilutions, serial dilutions, and then from each dilution you will spot on the plate, and then you can observe. And to calculate the activity, actually we need to take consideration which one of the spotted material don't generating inhibition zone. So this one will be our critical dilution, and as an inhibition zone, normally we believe that it's a 2 millimeters diameter. If it's less than 2 millimeters, better consider it as a negative. So 1 arbitrary units, it's actually calculated by the dilution, because we made it 2 times serial dilutions, and it's a number of the first dilution that not presenting. In this case we can consider it, let's say 7, and P, it's a volume what we deposit on the plate in microliters. Because remember, we deposit, some way it's written, yeah, it's here, but you cannot see it. 10 microliters we deposit on each place. But well, the result is in milliliters, so we have this P like a conversion factor, 1,000, to have from microliters to milliliters. No, now please take a piece of paper and let's calculate. Let's make practical things. Okay, let's, we can see like half of the cell, half of the room. We can consider that here it's no inhibition zone in number 4. Finish, no zones here anymore. So one of the group can calculate with number 4, another group can calculate it with number 7. So you're having dilution, it's 2-time dilutions on the exposition 4 or 7, multiply by 1,000 to have conversion from microliters to milliliters. How much will be the activity? How much?
Volume 10 microliter.
Volume is 10 microliter.
16,000.
Okay? And another group that is 7? Who calculated with 7?
1600.
But you're doing PhD, it's easy for you. Right? Well, you understand the logic. Okay, now let's make it more difficult. If you have the very strong bacteriocins, obviously you can have even inhibition zone here, attained. So you have two options to make more dilutions. You can dilute up to 16 times, or you can do it different way. You don't make a 2 times dilution, you make a 10 times dilutions here. So in this way, you need to take attention that D will be 10, not going to be 2, and then you can have few results as well. This is very important because if you just do the inhibitions on the diameter, can be big variations. And this is a problem because you pull a little bit more agar, you pull less agar. Your agar was 2%, your agar is 1%, your agar is 0.8. All this can interfere in the inhibition zone, in diameter. So very often it can give a wrong result. So making a critical dilution, you can always have a better result that you can compare with the other one. So now remember, when we was talking about the bacteriocin, we mentioned that they are proteins. And how you can actually confirm that really our substance that we have activity, it's really bacteriocin. So first point, we need to do to show that it's a protein. Okay, if you treat with proteolytic enzymes, very clearly proteolytic enzymes will destroy the activity, and you will know activity after exposure to proteolytic enzymes. So you can take any proteolytic enzymes that you have in the lab. Don't need to be specific list. You can check what left over from another project, what is available in some kits that you have in the DNA extraction. It's a proteinase K very often. You can use all this, but you need to check with proteolytic enzyme to sure that you destroy the activity. Now, fact that bacteria can produce the lactic acid, you need to be always sure that you do the neutralization of the supernatant to avoid the fact that some lactic acids produced. But the problem is coming when you have this class 4 bacteriocin, that complex molecules, bacteriocin plus lipid, bacteriocin plus polysaccharide. So you need to do some, let's say, alpha amylase, you can do some lipase as well. But then you can, depends on the results, you can say, okay, my bacteriocin is a pure protein because destroyed by the activity of proteolytic enzyme, but not being at all affected by alpha amylase, so good, it's not having this extra. And this part is important, so it's doing again, you have your cultures, you take a supernatant, you expose to, let's say it could be 0.1% of 0.1 milligram per ml, chymotrypsin, proteinase K, pepsin, any proteolytic enzyme, you culture, then you need to be sure to boil the sample for, let's say, 2-3 minutes only at 9800 degrees to destroy the enzyme, because enzyme is there, and you need to destroy to stop the enzymatic reaction. You spot on the plate in the same way like before, and then you serve. If you have the inhibition zone, it means proteolytic enzyme was not changing nothing, so it's not a protein. You're having something different from protein, could be diacetyl. If you have no inhibition zone, obviously this is bacteriocin. Of course, you need to have the controls. You put a pure enzyme, you put the denaturated enzyme, you can put non-treated with the enzyme to be sure that everything is okay. But then it's coming to another point. Okay, now you know you have the bacteriocin, you have something that is good, but now you need to be sure what is the identity of your bacteria. Because if imagine you isolate Streptococcus pneumoniae. Do you want to use it this time? No, I mean it's a pathogen. You want something that at least sounds like a safe, and then you can check the safety after that. Something that people was always doing in the, let's say in the 20 years ago, they was doing API test. BioMérieux, the company from France, developed this test. It's very easy because you're having 49 different sugars, and you have all the media ready. You just put your culture there, everything is good, only the problem is cost a lot. It's really expensive. And there's another problem. Imagine you have the strain Lactococcus lactis. Lactococcus lactis is a very interesting strain because he carries several plasmids. And several of the sugar metabolization genes are on the plasmid. But there is no guarantee that the plasmid will be there. Sometimes they have 3 or 4 or 10 plasmids, sometimes they have only 1. And if you lose some of the plasmids, that is not such important for the strain, for the living condition of the strain, maybe you lose some fermentation properties. So sugar fermentation property is a very good tool. It's already described very well in the Bergy manual, but the problem can be giving some errors. So normally how it's work, you have this media that is already provided. All the places except one is a control, no sugars. In all these places it's a different sugar. They are marked here on the file, but it doesn't matter really which one is where, but you have glucose, fructose, mannose, starch, whatever you can imagine, 49. And then you, and it's a pH indicator as well. And if the bacteria grow, since it's lactic acid bacteria, because this is made especially for lactic acid bacteria, then you acidify and the pH change. So obviously the first one needs to stay blue, because if first one change to yellow, you have something wrong. They're not supposed to be a growth in one. The rest can grow, here become black, from blue change to black because it's inulin. This is the way they work. But you don't have the strain that will give you this image, because it's like utopia. Then it's not existing bacteria that can metabolize 49 different sugars. But again, in the reality, you can have it like different colors, and some of them change, some not change, and then you have you enter in the website of the company, you put your sugar fermentation, they're telling you more or less what you have. The problem, this was a very good method till end of previous century, but these days not anymore, because the fact that it's not a constancy. This can give you idea. But why we can use this method? This method is very good to know what kind of sugar your strain is fermenting, because again, if you want to use it, let's say a starter culture in cheese. Cheese have what kind of sugar? Have lactose, and more or less is this. And obviously, if you have strain that cannot use the lactose, because one of these here is a lactose, you can see the strain is not metabolizing lactose. So how this strain can be applied in the cheese? There is no way. So, but this can, or another option, you want to produce your bacteriocin in industrial condition, and you want to use some waste products. But your waste product is very rich on mannose or maltose. So if the strain is not metabolizing mannose or maltose, obviously you cannot use this waste product for the growth of your strain. So it's giving you a kind of puzzle to know what you can apply the strain and how you can produce later. Obviously, biomolecular method is coming in the game, and then showing good options how you can actually use to optimize your work. Remember yesterday when I was isolating, we had we made it 3 times the same product, right? First group we used Dahi, second group we used, what was the name? Third group and fourth group was used pickles. So what is the chance to isolate the same bacteria? It's a highly possible, right? Because you're using the same product, the chance to have the same bacteria isolated in multiple occasions is a very possible scenario. So we need to do the RAP PCR or RAP PCR in a way to make a differentiation. So simply you isolate the DNA, you run the PCR, that is based on the RAP PCR and RAP PCR, they're based on the repetitive fragments in the DNA. And in this way you will generate a pattern that is a very same, if you have the same strain, you're generating the same pattern. So if you have the DNA template, the primer is only one primer in this PCR. They sit on the same, when they have the complementary sequence, they can sit in a different place, but they will generate the same pattern. And in this way, you can have the comparison. Obviously, this is with one primer, with another, with 4 different primers. And then if you look here comparing, like isolate 1, 3, and 4, they generate the same pattern. So most probably this is the same strain, isolated in multiple occasions. Because the same result, 1, 3, and 4, 1, 3, and 4, here it's not working to it, 1, 3, and 4. But no, it's working here in the bottom. but they is showing the same pattern. So obviously 1, 3, and 4, it's exactly the same strain, isolated in different occasions. So this is, I'm stopping now to talk isolates. Till previous step was isolates. Now I know 3 and 4 is the same strain, and I can continue only with one of them. So you already, by REP or RAP PCR, you can actually eliminate repetitions that you had in your work. Now it's coming the next step. We need to know what we have. Well, we can use the species or genus-specific primer, but again, this method is already in the past. This day we have some better, but again, historically, this is the next step. We can run the genus-specific primer that you can confirm the genus of your strain. Remember, I was mentioning yesterday that enterococcus is quite difficult to do the 16S. But so if you can do the genus-specific primer to detect this enterococcus, it's already okay, because you can combine it with the biochemical and physiological test recommended by Bergy, and you can actually find what kind of enterococcus you have. But you can use the species-specific primers that will generate a specific size or specific amplicon. Interestingly, like in 2005, 2006, people was happy with these results. You have the species-specific primer. Bingo, I have Pediococcus pentosa cells. Positive control give the same, strain give the same. The problem is you have the same size of amplicon, but sequence could be different. So now, next step, you need to sequence this amplicon to confirm that the sequence that you're looking for, that is your target sequence, in fact is the really one that you have it. So this is a short explanation how the PCR works, but I presume everyone already knows this. It's okay? Must be, because I mean this is in the basic courses of genetics, and some should be very upset if you don't know. So you sequence, but now it's coming the next point. We're doing the 16S. Next step that is become. In the beginning, 16S was considered as a problem, expensive, not too many places. Now, most of the labs, they have these options in the campus. Very often, it's options to have it this or the next city. It's not really a big deal anymore. And you have performing the 16S rDNA, or you're performing qPCR, you have the segment. It's if it's possible to have as big as possible, like 1,000, 1,500 is better. Some primers detecting smallest fragments, about 500, 600, but biggest is, in this case, is the best option, because you can be more better when you do the blast. You do the PCR, you purify the product, you give for the sequence, or you do it yourself, and then you blast, and then you have your results. Again, Pediococcus pentosa cells confirm it in the case of the different strain that we have, because we have two, remember, two strains from this product that we isolated. Was giving enterococcus, but again, enterococcus faecium, 98% is, was not happy, because these same results can appear with enterococcus fecalis, with enterococcus munti, with enterococcus caseoliflavus, enterococcus cariae. Enterococcus 16S is not a good option, because it's very conservative region, and very same region for the different enterococcus. So it needs to be combined with the solutional method. Well, in the last year appearing another method, it's Malditov. It's again a method that actually has several critics about this method, because method is not difficult. You have a pellet of your culture, you have the strain colony from the plate, you put on the plate of the equipment, then you put in the machine, then it's heating, they was doing evaporation of all the material, detecting the spectra, and based on the spectra on the proteins, comparing and doing analysis and telling you what species do you have based on the protein profile of detected in this process. The problem, this method is very good for the industry because it's very quick, but the problem in this method can be very approximative. You see enterococci, you cannot even distinguish them on the 16S on the genetic level. Imagine distinguish them on the protein level of the surface proteins. So for the industry it's perfect, because in the industry you tell them, listen, you have salmonella in your sample. It's already enough for them to have a big problem in the industry. But in the laboratory, when we know exactly the specific specificity of each strain, not just genus, Malditov could be a very kind of general approach, but not a really detailed. Imagine how we can distinguish different strains on the same species just from the protein level. This will be very difficult. If you think about Lactobacillus plantarum, Paraplantarum, and Pentosus, they're difficult to be distinguished on genetical level sometimes, and only on the protein level you can make this work. Not really. Well, Malditov not going to be happy with my comments because they want to sell their machines. But in the same time, Malditov, the companies, actually Bruker, Biomer, or the rest of the companies producing this equipment, they went for the new generation because they realized that their equipment, in fact, is not so sophisticated to meet the scientific exigence. They meet industry, but not on the academic level. So they made it a new addendum to the machine that they have the Fourier transform infrared spectroscopy. So once they have the classical Malditov, they have the spectroscopy second section that actually is helping to do better distinguish, and including then some of them have even a Rabotyper that helping to really find what do you have. Only the small problem, this one cost $2 million. Well, even in the very healthy labs, this is a lot of money. But this is at the same time, I think it's some strategy that several universities have. They have these multi-user facilities. They're buying equipment for entire university. Sometimes even two universities buying the same equipment to use it by two of them. Because this kind of equipment, such a price, I mean, you need to combine with your neighbors. You cannot just have it in your lab. You need to do it as a central facilities of the university. They will serve several faculties, and this way it's work. Because this kind of equipment, if it's work, it's okay. If it's standing, stopping in the corner, it don't generate nothing. It needs to work. And in my lab, I'm not going to do the identification every week. I'm doing like 2-3 times per year when I have new collection, new strain isolated, and they need to do so. Obviously, the central analytical facilities is a good option for this. But it's something that is work quite quick, quite easy. But again, even with this identification, I will go for the full genome sequence. Because these days, full genome sequence become a compulsory point as well for the identification. If you want to use the strain as a probiotic, you're going to use as a starter culture, you need to do the full genome sequence. There is no option. Several agencies, to order to certificate your product, they ask you to provide full genome sequence. They want to be sure that you have sequenced your DNA total, you made it appropriate bioinformatic analysis, and you show that the strain don't have the dangerous parts in the genome, because again, safety is priority. You don't want to introduce something that having genes that could be related with some dangerous feature. Well, now you know that you have the bacteriocin, you know the identity of your bacteria, and you need to think about how you can characterize your bacteriocin. First of all, you need to be sure at what level your bacteriocin is produced and where you can stop the fermentation. Okay, in the laboratory, if we do the 24 hours, it's not a big deal. But in the industry, if you can stop the fermentation earlier, it's mean money, because imagine in the lab we use the lab tube like 10 milliliters, so we're using the RMR flask with 100 milliliters, but in the industry there will be 20,000 liters. You don't want to heat this huge bioreactor for a longer period. So obviously you can do a very simple way. You can do the growth production study. So you just grow your culture, you're taking every hour samples and you measure optical density, because it's easy to just to do the plating and do the cell count, but in certain extent the optical density gives you a good idea about the growth. So you're growing your culture, you see it's reaching the stationary phase more or less 16, 70 hours, even 15, and then staying in stationary phase. In other cases, reaching a little bit earlier, about 10 hours or 11 hours, reaching the stationary phase and go there. Then we do the acidification as well, because on this way we can understand something about the structure of bacteriocin, because sometimes bacteriocin, when they go to the low pH, they can make a clumps. They go together. two of them, they will, two molecules of the protein, they go together, they go four, and very often when they make these clumps, they are not anymore active. They're losing their activity. But this is normally associated with low pH. So it's important to know how the pH acidification going, and as well very important to know what's the level of the bacteriocin production. So in this way, you can see we was doing a fermentation for 48 hours, for 2 days, but in fact, after, let's say, how much is it, 21, it's staying stable. Why you should go for 48, even in 21 I can stop it? Don't make sense, right? Okay, for the lab, if you put it on Friday and you want to work on Monday, it's already too late because it's going down. But I mean, you made it a culture in the let's say in the 8 o'clock in the morning, but then you had a meeting and you stop it more at lunchtime, okay, because you're in this area that you already have the maximum of production. But again, you need to know where is the maximum so you can stop it. In the second case, in the bottom, you see have a very big peak in the end. But this makes sense to have this high activity, because activity here as well is high, because it's related for like, this will be 2 with 8 zeros in the back, activity. It's quite high. You have more here, 1 with 9 zero. So 1 zero more, 1 log more of activity really make a difference, but make difference on the energy use and the time. So I can rather stop again on 21, 22 hours here because I have enough and you don't need to go too much. And here as well you risk to go down because making these clumps. Then is another problem. Okay, here it's showing like stable optical density, but could be a little bit kind of not true. Because obviously here you have dead and live and dead cells. In stationary phase you have live cells, you have dead cells. The problem with optical density, you're not detecting live or dead, but if you do the plate count, you can see that actually start to declining here. If you do the cell count, not just optical density. And when you have too much dead cells, what's happened? That cell is lysing and they liberate proteases from the inside. So some proteases could be available in the end of the process, in the end of the stationary phase, and they can interfere with the integrity of your bacteriocin. Then you know what is the time, you know what, how long you need to take a fermentation to have the bacteriocin production. So you know where you can stop the process. Again, this experiment is done in 37. But if you work with a probiotic for different kind of targets, poultry or fish or even agriculture, you need to adapt to the temperature that is appropriate. But now it's coming another problem. Lactic acid bacteria, what is their growth temperature preferences? More or less, Enterococci below 15, they don't like it cold temperature. Lactobacilli below 20, it's not so easy. Going up more than Streptococcus thermophilus, 43, 44. But if you go higher, they will be not happy. I mean, they have their preferences, and we need to respect their physiological needs. So now, you're going to do something for the crabs. Crabs, normally they grow in the very cold water. So obviously, lactic acid bacteria are not going to be a good potential probiotic for the crabs, because you need something that will be happy in cold temperature. So you need to look for the different group of microorganisms. And this could be archaea, this could be some bacillus, because bacillus, they have larger temperature preferences. So again, lactic acid bacteria cannot solve all the problems, because again, you go to the living organism, and this living organism has specific temperature, and lactic acid bacteria has specific preference for the temperature. So we need to match together in order to work. But again, we need to find now how this bacteriocin is stable. Because if it's not stable, it's not going to work. Imagine you having bacteriocin that is not stable in low pH. Can you use it in dahi? Dahi, what's the pH of the dahi, more or less? I presume it's similar like to this on the yogurt, presume. It will be like 4 something, called 3.9, 4 point something, more or less in this area. But if the bacteriocin is not stable at 4, they're not going to work in this kind of product. So you need to check the effect of pH. You just take a supernatant. Please, when it needs to stop, you can let me know. Okay, because in Brazil, normally I'm used to do